All right, our next speaker, who also, I honestly don't know why people say yes sometimes to me, but he did. I sent an email pleading, and within like 10 minutes, he was like, I'll do it. I was like, my gosh, I would have thought about it more, but okay, thank you. So, Prayag Murawala is an assistant professor at MDI Biological Laboratory, and he's at the Clinic for Kidney and Hypertension Diseases at Hanover Medical School in Hanover, Germany. I'm just going to do another quick plug here. MDI Biolab is grossly unknown as far as I'm concerned in Maine. They do extraordinary work. All of these guys, if they haven't heard it yet, they will before they leave. I fangirl all of the work that they do. Sometimes like individuals, sometimes the whole organization. I think MDI Biolab is one of these places. I would imagine in Germany, you essentially walk on water. And here in the States, we hardly know who you are. So it's a shame, and we really need to fix that. Okay, I'm off my soapbox. Prayag studied across India before settling for his graduate studies in Pune, India? Pune, yeah. Sorry. He then moved to Europe, where he studied at the Max Planck Institute of Molecular Cell Biology and Genetics, and then moved from there to the Center for Regenerative Therapies in Dresden, to the Research Institute of Molecular Pathology in Vienna. I just like the fact that you're so well-traveled. That's why I included all this.
Thank you.
After exploring Europe, he saw the light and he moved to the East Coast of the United States to start his lab at MDI Biological Laboratory. He currently holds a joint assistant professorship, as I mentioned, here and in Germany. And his pop culture reference was he loves science fiction movies, particularly related to space and astrophysics. And the examples were Interstellar, Gravity, and The Martian. And for anybody who wants it afterwards, he also sent me a clip from one of the Amazing Spider-Man movies that is related exactly to his work. Really enjoyed that. Don't have time to show it. So, all yours.
Okay, hi, good evening. So the topic that I'm going to discuss today relates to all of us. And that is, can we regrow our body organs? And the reason is, as we grow old, our body organs fail, or we suffer an accident and we may lose, for example here, a leg. So the question is, can we regrow them as it happens with some animals? For example, here I'm showing you an animal called planaria. You can cut this animal into 3 pieces, and all 3 pieces, the head, the torso, and the tail, they will make 3 new organisms. You can also cut this animal vertically, a little bit on the left side, diagonally, triangle, square, and all of these pieces will make a new organism. While this is a fascinating example of tissue regeneration, this is not the only animal. There are many animals that can do tissue regeneration. Probably you know a lizard can lose its tail and it can regenerate those tail. You can cut a leg of a starfish or a cockroach and it will grow back. You can cut earthworm into center and it will make 2 different earthworms. While all these organisms are known to do tissue regeneration, my favorite is this particular animal, which we call axolotl. It's a salamander, and it's a champion of regeneration. The reason we call it a champion of regeneration is because it can regenerate pretty much any body part in its body. It can regenerate its legs, it can regenerate its tail, heart, liver, lungs, spinal cord, and even brain. So you can imagine why we love this organism. Now, among all these organs, the specific organ that we focus in my lab is limb. Limb is a complex structure made up of many different tissue. You know, you have skeleton, muscle, etc. And a scientist can cut a leg, and similar to us, they also bleed. And after bleeding, they can heal this wound. Now, once the wound is healed, that's where human body stops its growth. But this animal is special. It can activate the cells that are underneath, and these cells will then go on and form a structure, which is a conical shaped structure that we call blastema. Now, blastema contains all progenitors that know how to reconstruct an entire limb. So they go on and make a limb. Now, as you can see, this process, although looks fascinating, is extremely complex because there are many different cell types who have to talk with each other so that they can make a fully patterned limb. Now, complex question also requires complex methodologies. And in my lab, we do genome engineering to label an animal in a way so that you have individual cell type labeled with different colors. For example, you can see the peripheral nervous system labeled in green, central nervous system labeled in red. Here I'm showing you another example where you have a fibroblast labeled in the limb or a skeletal elements labeled in the limb. Now, once you have this kind of a labeled environment, what you can do is you can separate these red cells from rest of the body, and then you can pick one cell and ask what kind of genes are expressed in this cell. And I'm not talking about looking at one cell, but nowadays we can take thousands of cells and then we can analyze what are the gene expression in thousands of cells. The method, which is called single cell transcriptomics, allows us to interrogate what are the gene expression changes that happens in the course of limb regeneration. Now, using this kind of a transgenic animal and this kind of a complex methodology, one of the major findings that my lab has shown is that the fibroblast cells in these animals, they can go back to their embryonic identity when the limb was developing. And then, so the process we call dedifferentiation. So using dedifferentiation, they acquire the embryonic identity, and then they can go on and make different compartments of the limb. And this is what happens in salamander, and we cannot do, and that is why we cannot regenerate. So I'm pretty much at the end. So I would like to wrap up my talk with this famous quote by Richard Goss, who worked at MDI Biolab in '69. If there were no regeneration, there could be no life. If everything regenerated, there would be no death. So with that, thank you, and I'm open for questions.
Watching that cool graphic where the lizard gets his limb cut, it reminded me of the ship of Theseus and the paradox of, I wanted to focus on that limb, it just went off screen.
Sure.
The blastema, like how does life sort of solve that problem of which part to regrow? I mean, could it technically regenerate from just that leg, and why not?
It's a very fascinating question. So in the body, in any organism's body, there are cells that know their position. So in our scientific language, we call it a positional memory. So not just, just within the limb, just to explain you have upper arm, lower arm, and hand plate. And cells across this axis, which is called proximal to distal, they know their position. So if you cut here, they will only make fingertip. If you cut here, they will make lower arm plus the hand plate. If you cut here, they have to make upper arm, lower arm, and fingertip. So the cells know their position, and it's coded via a certain cluster of genes, which we call Hox genes. So this is how the cells acquire positional memory, and then they use it for the patterning information.
Thank you.
My pleasure.
My question's a little related to that idea.
Sure.
So you had mentioned that the embryonic cells that start out, but then if there's a mutation in the hand as it's growing out, let's say, and there's additional finger or something happens environmentally, do you consider it the same limb?
So that's a great question. So the external stimuli can certainly influence how the blastema is growing out. So if you have a certain chemicals, for example, sonic hedgehog, it's a molecule. So that, if it's present in the system, and if a limb is regenerating, then instead of forming 4 fingers of the forelimb, they can form many fingers, like 7 or 8 fingers. So it can happen.
Based off the research that you've done so far, do you think it's possible for humans to develop or have something like that happen, or is it too far out of the realm of possibility in your mind?
This is the one I always get every time. So, great question. For me, the more realistic aspect, which I did not discuss here, is the fibrosis. When we get an injury, we develop fibrotic scars, whereas these animals don't. And it's the same cells that are responsible for regeneration are also responsible for fibrotic scars. So the likely outcome or the immediate future that I see is that, if we can understand it thoroughly, then at least we can reduce or probably eliminate fibrotic scars in mammals.
My question is related to that. I was wondering if we could possibly take the stem cells of this creature and put it in a human, and if that could help. But yeah.
That's a nice question. The only problem with that is, instead of growing a human limb, we will make a salamander limb.
What is it that makes human cells so different from that of the lizard? That they aren't able to do that?
Yeah, so that's $1 million question, basically, and that's where government gives us money to so that we can continue on the bench. The major difference, as I mentioned, is that the cells of salamander can go back to embryonic identity, whereas our cells cannot do. Now, that's where the question comes. What is so different between salamander cells and our cells? They have fibroblasts, we also have fibroblasts. Why those cells can do it and why we cannot? Now, certainly, there is also a 400 million years of evolution between salamanders and humans. So although genes are same, or they are similar rather, I would call it, there are differences. And then the question is, what are those differences that govern this ultimate outcome that is differing between these two organisms? And that's what we are doing in the lab, essentially. Thank you. One more.
Hang on. So you made mention of the fact that the cell can revert to the embryonic state. A cell in an embryonic state during its initial formation is in a much different, it's in an egg or it's in utero, it's some other form, versus regeneration outside ex vivo. So is there signaling from the environment or the local environment that changes that growth pattern?
Definitely.
5 seconds.
It's both intrinsic and extrinsic. That's the simple answer. We can discuss more later. Thank you.